Historically, drug discovery has worked much like searching for a needle in a haystack, testing one hypothesis at a time, hoping eventually to find the right biology. But what if, instead of searching for one hypothesis at a time, you could systematically map the entire haystack? For more than a decade, Recursion has been building one of the first and now one of the largest AI-native data factories, continuously generating biological data, building foundation models, and experimentally validating the predicted insights in a closed loop. These discoveries are then tested, refined, and validated in our labs before being fed back into the system, creating that recursive learning loop between biology and AI, where every experiment strengthens our models. Every model informs the next experiment, and every cycle deepens our understanding of human biology. So today, we'd love to take you behind the scenes. This is not a black box. This is real science and tech in action to show you how that engine works at Recursion. Let's start with the cells, lots of them. First, to study a disease, you have to work in cells that actually resemble it. When we started this in 2021, this was unprecedented. Human neurons at this scale, editing every single gene, imaging all of it, there was no blueprint for this. The cells are grown from stem cells and differentiated into neurons over weeks, using specialized proprietary protocols, developed in partnership with Roche Genentech to achieve this necessary scale. Producing these cells at scale is extraordinarily challenging. Keeping iPSC-derived neurons alive, consistent, and biologically representative requires overcoming significant batch-to-batch variability. And fun fact, to build this genome-wide map, we had to create over 1 trillion neurons, roughly the equivalent of 12 human brains worth of neurons.
Second, biology is dynamic, so you need the ability to manipulate cells precisely and measure changes in their cellular state. This is the part that turns us from just a cell factory into a data factory. Recursion has built the tools to systematically manipulate these cells at the genetic level and quantifiably measure what happens next. Here, you can see Recursion's Echo acoustic liquid handler installation. 2.5 nanoliter droplets of CRISPR-Cas9 reagents are transferred by acoustic pulse into these 1536-well microplates. Recursion scientists have developed dozens of ways to manipulate cells at this nanoscale so that they can do it up to 2 million times per week. Importantly, our labs are specifically designed to intentionally randomize these experiments so that the machine learning models have to work hard to understand what changes are the result of real biology instead of experimental design. Lastly, biology is nuanced, so you need the ability to tease apart subtle differences in what's going on within the cells. Low-dimensional data simply won't do. These same cells go 2 places. Here, every cell is imaged, and then we have the capability to sequence those cells. To our knowledge, no one else does this at this scale. It can yield thousands of measurements from a single experiment. This high-dimensional data is the raw material our foundation models learn from. And because it's this rich, the data is reusable, mined again and again across programs, rather than generated once and thrown away. It's also future-proofed. As AI continues to advance, this same data set can be revisited with new models and algorithms that don't even exist today, allowing it to generate new insights from the same data to reveal discoveries that today's AI may not yet be capable of seeing.
The data is used to train our models. The models point to the targets. That's lab in the loop. Every image, every sequence of every genetic perturbation or otherwise we run in the labs comes through BioHive 2, Recursion supercomputer ranked among one of the fastest in the world. Here we train state-of-the-art foundation models to create a unique embedding or fingerprint of every cellular perturbation we've ever performed, whether that's on images or other modalities. Our genome-wide images are processed by these foundation models into a single large data set, a map, giving us a novel view of biology. Like a criminal database, some of these fingerprints are known to be associated with bad actors or disease-causing mutations. By comparing these known fingerprints with other fingerprints that were created in our lab for their maps, we can find other genetic manipulations that create similar effects. These connections create new hypotheses for what might drive or rescue a disease state, pointing us toward a potential novel discovery.
A hypothesis from a model isn't a discovery yet, so it goes back into the lab, the same loop running the other direction through a validation cascade that gets progressively more demanding and more specific to the disease. That's the loop closing, an AI-discovered hypothesis de-risked at the bench against the same high bar our pharma partners hold their own programs to. The biology the model predicted is the biology we saw at the bench.
Much like our approach to biology, we solve complex chemistry problems through a feedback loop between experimentation and computation. Across our programs, we've advanced quality, novel candidates after synthesizing only around 300 compounds, roughly 90% fewer than the industry average. Here's what that actually looks like in practice.
First, before a single molecule is synthesized, the team works through the decisions that shape that cycle. We have hundreds of machine learning and physics-based models integrated into a single drug design environment called Centaur. Starting from the target properties, we can iteratively design novel molecular structures for biological targets, an approach known as generative de novo design. The design cycle feedback loop automatically updates our ML models so that they improve with each design cycle. We have seen that our AI native product engine performance is repeatable. For example, we have produced over 10 development candidates. But it's not just about quantity. Each of these candidates reflects that same discipline and that same high bar we hold our biological targets to. That focus on quality and novelty over volume is what we believe will define AI's true impact on drug discovery and development. On our AI native product engine, a chemist can have a full design cycle up and running in about a day, which would be nearly impossible without the modularity, data, and AI approaches built into our engine. Most drug design leans on decades of precedent, bespoke experiences, familiar chemistry, and binding pockets. Instead, our engine increases the aperture of the chemist to explore novel, often complex chemical space efficiently by leveraging tools that effectively analyze complex data, forecast non-obvious design hypotheses, and predict how to synthesize even where nothing like it exists yet. For example, binding affinity. How strong a molecule binds its target is one of the earliest signals of potential scored with 3D structures from models like NESO-1. From there, hundreds of models, including ADME, potency, advanced simulations, and AI retrosynthesis, are run by the team as one integrated system. That narrows millions of molecules down to a short list that's synthetically accessible, not just promising in silico, but actually makeable, before a single one is sent to the labs.
Next, these designs move into our automated DMTL labs, where we make and test them in the same lab-in-the-loop approach running once again. This is only possible because we control the design, make, and test steps within one connected system, rather than handing off between disconnected tools and teams. Here, you see Recursion's ChemSpeed platform, performing complex, multi-step medicinal chemistry, not just simple one-pot reactions. Automated workup, purification, and digital sample tracking manage thousands of compounds while creating a digital record of every experiment. Our in-house AI agent helps scientists plan synthetic routes and maximize the productivity of both the platform and the chemist using it. Once synthesized, compounds move directly into automated testing. Scientists can describe an experiment using plain language, and the platform generates the laboratory protocol behind it. No manual coding required. Such automation has reduced our pharmacology testing cost to roughly half of the traditional industry benchmarks. But the real advantage isn't just speed and cost. Every result feeds straight back into our models. That's what lets us reach a development candidate after synthesizing around 90% fewer molecules than the industry average. We're not handing off between prediction and synthesis, we can make it right here. That's why we've put so much research and development into closing this loop. Not to make more molecules, but to make fewer.
Once a molecule passes the high bar to become a clinical candidate, it re-enters our AI-native product engine specifically for clinical technology, or ClinTech, designed to bring the same data-driven approach we built for discovery into every stage of clinical development. First, before a single patient is enrolled, we need to understand who most clearly benefits from this medicine. Many late-stage trial failures occur because studies enroll broad, heterogeneous populations where the real treatment effect gets diluted. We use AI models like Cardinal to bridge the gap between the laboratory and real patients. First, we learn which cancer cells respond to a new medicine in the lab. Then we use Cardinal AI to compare those cells with thousands of real patient tumors to identify patients whose biology looks most similar. That allows us to predict who may be most likely to benefit and why before a single patient is enrolled in the trial. In one analysis, this approach identified a specific genetic mutation that consistently distinguished tumors predicted to respond from those that were resistant. In other words, we discovered a potential biomarker simply by connecting laboratory biology with real patient data.
Second, once we know who to look for, we have to find them and get them enrolled. And this is where trials most often stall. Nearly 80% of clinical trials experience delays, and enrollment is the single most common cause. Our Site Finder tool draws on real-world data from more than 300 million patient records and intelligence from hundreds of thousands of trial sites worldwide. So we can identify high-quality sites and investigators in hours instead of months. We apply the same inclusion and exclusion criteria the trial will use to see exactly how many eligible patients exist at a given site, and choose sites with strong track record and low competition from other trials. That combination has improved enrollment rates by 30 to 60%, cut site and country feasibility assessment from months to hours, and taken up to 3 months off study startup timelines.
Third, once a trial is running, decisions have to be both fast and rigorous. Every scan, laboratory measurement, biomarker, and clinical outcome generates new evidence about safety and efficacy. We continuously integrate these signals with longitudinal, real-world data to understand how outcomes compare with the disease's natural history and with similar patients receiving today's standard of care. This real-world evidence can help us contextualize early clinical signals, refine development decisions, and build a clearer evidence package for regulators, physicians, and payers. For example, our ClinTech biometrics engine replaces the manual, per-trial analyst workflows that most programs still rely on with a single unified data infrastructure. Dose escalation decisions that typically take 7 to 10 business days come back in 24 to 48 hours, about 7 times faster, while data to decision time drops by 60 to 80% per review cycle. We're already applying this approach across our pipeline. Together, these tools help us select the right patients, find them faster, and interpret emerging results in the context of both the trial and the real world, creating clearer signals and enabling faster, more confident decisions.
That's what an end-to-end system makes possible. Not one better medicine, but a renaissance in how they're created in a systematic and repeatable way. What we have at Recursion is a learning system, a truly recursive system that discovers novel causal biological insights, designs drug-like molecules at a fraction of the time and cost of industry, and very importantly, connects that patient data to select the right patients who would most benefit from our medicines. Increasingly, AI agents are also working alongside our scientists across the entire engine, from mining these maps at unprecedented scale, from planning chemistry synthetic routes, and from setting up the right trial sites so that we can recruit patients at a pace that they would be proud of. Being AI native from the get-go is what makes this possible. This isn't just an idea or a rendition on a slide. This is a product engine in practice that has already put multiple programs in the clinic and is starting to change the way medicines are made today and in the future. Better medicines built faster for the patients who are waiting.